Okay, so today I'm going to be talking about glucose metabolism, which is a bit of a shift from the other talks today. We are interested in glucose metabolism in the GRIN1 mouse models for a few reasons. First of all, we've been studying the GRIN1 knockdown mouse, which is just a mouse that has low levels of GRIN1, GLUN1 subunit. And we've seen a lot of evidence that they have an upregulation of the enzymes for glycolysis and for mitochondrial respiration. And this made us wonder whether of course, they probably have increased energy demands in their brain, and whether they are getting sufficient energy for this increased demand that comes with trying to compensate for the reduction in NMDA receptor function. And through our studies, we also studied the ketogenic diet to look to see whether this improved some of the phenotypes that the GRIN1 knockdown mice have. And we found that both ketogenic diet and exogenous ketones like beta-hydroxybutyrate could improve many of the phenotypes of the GRIN1 knockdown mice. So this is really the reason why we started looking at this. I mean, in addition, of course, we know that there are GRIN patients who benefit from a ketogenic diet, and we also know that GRIN patients are often underweight. So it's worth looking at the energy and metabolism of the mice to try and get some insight into what might be happening in the patients as well. So the mice that we study in the lab, today I'm going to be talking about this first model, which is the GRIN1 knockdown model, and it's homozygous for a mutation that doesn't occur in humans, but they have a really dramatic reduction in the amount of NMDA receptors that they have. We also have two patient variants, which we study in the heterozygous state, like the patients, and one of them is a loss of function, and one of them has mixed effects. I'm not going to really be talking about those today, but we are doing some of these same studies that I'm going to describe for the knockdown mice in these patient variant models as well. So the first thing that we wanted to establish, we've known since we've made these mice over 30 years ago that they have lower body weight. And so you can see this, and now it's divided between males and females. I'm going to make sure, okay. So you can see that the males and the females both weigh less, and the males in particular have a reduced body weight. We looked at food consumption and saw that it wasn't really different in the males, and there were some reductions in the females, but in general, the amount that they're eating doesn't really explain their reduction in body weight. Another thing that we did was something called a DEXA analysis, where you're looking at the amount of the body that is fat or lean or mineral, the bones. And what we saw is that in the mutant mice, both in the males and in the females, there's less of the fat and more of the lean tissue. And there's also slightly more mineral. And that's really because so much less of their body weight is in fat. So they have a reduction in fat, which I think any parent would say also that their children also don't have a lot of fat on their body. Okay, so the next thing that we did was to look at some of the metabolism using something called a metabolic chamber. And this is where a single animal is placed in a chamber where you can measure the amount of oxygen that's consumed, the amount of CO2 that's produced, the amount of heat that's produced, and of course, how much they're moving around. So over here, we can see how much the mutants are moving around. And here we've combined the males and the females. And through the light cycle, you can see that they're more active, when the mice are normally sleeping, they're more active, but also when the animals are normally awake, they're more active. So they're active just throughout the light cycle, they're more active. They're producing slightly more heat, but really not that significantly different. The interesting differences were in the oxygen and carbon dioxide consumption. So here you're looking at the amount of oxygen they consume and the amount of carbon dioxide they produce, and you can see that it's higher throughout the life cycle. And this is actually a way that you can tell not only do they have a higher metabolic rate, but also you can tell what is their source of energy. And you can do this from looking at the ratio of the amount of carbon dioxide that they're producing over the amount of oxygen that they're consuming. So if this is a ratio of 1, then that means that they're using carbohydrate primarily as their energy source. But if they have one that is 0.7, that means that they're using fats as their energy source. So the fact that these animals have a lower ratio here would indicate that they're preferentially using fat as an energy source over carbohydrates. Even though they're eating the same diet, they're not using the carbohydrates as much as they're using the fat that's in their diet. Okay, when we were looking at this preference for fat as an energy source, it made us remember a paper that we had published a few years ago, where we looked at the effects of either supplementing a diet with omega-3 fatty acid or giving the animals a diet that lacked omega-3 fatty acids. And so what we saw was that there was this striking mortality that occurs when you remove omega-3 fatty acids from the diet. So, so this is the normal diet, the supplemented diet, the normal diet in dashed, the supplemented diet in the solid lines, and you can see, and the deficient diet that the males, over half of them died with this diet before they were even tested in behaviors. And we also saw this early mortality in the females, but it was really more striking in the males. So I think you will see this recurring theme here where the males have a more severe phenotype than the females in really every aspect that we've studied so far. I wanted to show this diagram to kind of point out a few of the hormones that are important in regulating glucose. So the two hormones that regulate glucose levels in the blood are glucagon and insulin. So when we eat, so after breakfast, we will have a higher level of blood sugar, and then this will stimulate the pancreas to release insulin. And insulin is supposed to then take that glucose from the blood and put it into tissues where it can be used for energy. And also, if there's extra, put it into the liver so that it can be stored as glycogen. Then when we are hungry, this low blood sugar will actually stimulate the pancreas to release glucagon, which will then liberate glucose from the liver so that it can go back into the bloodstream and normalize our bloodstream glucose levels. So they should stay within this really narrow range. So we looked at glucose in the animals, and the first thing that we do is something called a glucose tolerance test, where you give animals a big bolus, a big amount of glucose all at once, and then you take their blood at different intervals and you measure how long it takes to bring it back to the baseline level. So this is done when the animals are fasting here. And so you can see that even at fasting, the males have a reduced level of glucose. And then when you give them the glucose, their blood sugar levels increase, and then they go back down. So this is showing the clearance, and it's actually faster in the males than in the mutants than in the wild types. And this is true also in the females, but less of a difference in the females than it is in the males. Okay, the other thing that we then look at is what are the levels of insulin. And so again, looking at fasting levels of insulin, we can see that the fasting levels of insulin are actually lower in these animals. And when we give the animals insulin and measure glucose, so remember, insulin is supposed to take that glucose out of the blood and put it into tissues or put it into the liver. So the insulin is responsible for this drop in glucose. So when we give insulin, you should see a drop in glucose, but you see a really dramatic drop in the mutant animals that's even greater than what you see in the wild type. So they're very sensitive to insulin. So, you know, they don't have a lot. So when you provide insulin, you have a more dramatic response to the insulin. And in some of the patient variant lines that we've studied, it drops even further to the point where the animals can have seizures. Okay, so again, it's interesting when we thought about the literature and other studies that have been done. We remembered this study that was done on the serum levels of animals that have the same mutation. And they did have really significant reductions in a similar hormone to insulin, the insulin-like growth factor. In addition, they had increases in the amount of glucagon in their serum. So this was just another piece of evidence that we had going in that there were probably going to be changes in glucose homeostasis in these animals. Okay, so in addition to changes in glucose tolerance and insulin, induced glucose clearance, we also saw a difference in exercise-induced glucose increase. So what you should see when you exercise an animal is that the basal levels of blood sugar should increase. So activity is another thing that will cause that liberation of stored glycogen so that you get glycogen turned into glucose and in the bloodstream. But you don't see this in the mutant animals, so there's not really this same increase, and in some animals you actually see a decrease in blood glucose after exercise, which you really should elevate. And not surprisingly, the animals also, when you put them on this treadmill, they kind of exercise to exhaustion sooner than the wild type animals. So, of course, I mentioned that both insulin and glucagon are liberated from the pancreas, and they're actually produced in these islets here, which you can see in these sections of the pancreas as these kind of white circles in this field of purple. And we saw that in the males that these were very sparse and that they were smaller than in the wild types. And this was not really seen in the knockdown females to the same extent. So you can see they're similar size. And so we did this study looking at just the size of the islets, and you can see that it's really reduced in the males and not as different in the females. But I think you can also see that in general, wild-type males have larger islets and more islets than females, than wild-type females. And you can see this also in this slide here. So in these sections, we've stained them for insulin in the green here, and for glucagon in the red, which should make like a nice ring around these. So you have beta cells that are producing insulin, and the alpha cells are producing the glucagon. And here you can just see in the whole pancreas where the number of islets and the size of the islets, and you can see in the male that they're much smaller, and you don't really see this difference in the females. So there's a really striking difference in the males in terms of the number of particularly their beta cells. And you can see this here, that the beta cells in the males are reduced, and they're kind of similar to the levels that you see in the females. The alpha cells were not really different, and if anything, there were more alpha cells in the females than in the males. Okay, so I think I'm just going to skip this slide because it's just trying to understand what might be happening, and we think that this might be due to actually the way that these islet cells are produced in development, and it requires innervation from the sympathetic nervous system. And I'm going to end with this summary slide saying that the knockdown animals have reduced weight with less fat and more lean body mass. The metabolic chambers indicate a greater use of fatty acids as food energy source rather than carbohydrates. The pancreatic islets are smaller, fewer in number, and there's some disorganization. I didn't really go into that. There's lower insulin and glucose levels in the serum and a faster clearance of glucose, a higher sensitivity to insulin, and this is particularly seen in the males. Also, exercise does not increase blood glucose as it should. Some of the questions that we have is whether this is seen in patients, if it's a developmental phenotype where, do we see this from very early in development? Can this be corrected? And could these be useful biomarkers for clinical trials? So I'll end with thanks to the lab, particularly Tatiana is the one who did all of these studies, and to the collaborators and funding.